I'm Dr. Khokon Kumar Dotto. My Christian name is Dr. David Moon. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Discovery of mature microRNA-34a inside the cellular nucleus and the importance of nuclear microRNAs. I was lucky to get the opportunity to pursue my PhD research in the School of Medicine, Kyoto University in Japan. One of my research projects was with microRNA-34. Some other researchers were also involved in the project. The environment of our laboratory was a little bit like a team of researchers. All the members of the laboratory tried to help one another as much as possible. I had co-workers in the laboratory from different other countries. But a senior doctoral researcher in the laboratory was the best help for me. He was a Japanese student. Interestingly, we were awarded PhD degree in the same year. He is now working as a faculty in the Nagoya University in Japan. A part of my project was to localize and to visualize microRNA-34a inside the cells. It was a very difficult attempt at that time. To our knowledge, at the starting of the project or experiments, nobody tried to do this type of experiments in mouse, rats, or even in humans around the world. Mature microRNA-34a, microRNA-34b, and microRNA-34c are very small RNA molecules. They had a very little differences in their sequences, which made them very difficult to localize and to visualize separately. But we received great help from our Danish company Hexagon. We could collect very specific probe mercury for the in situ detection and visualization of mature microRNA-34a. The probes are very expensive. I could collect that very easily because of the deep interest of my supervisor in the project. We prepared the rat tissues in the laboratory ourselves. We collected human tissue samples from South Korea. They were also very expensive. All the arrangements for a single experiment were economically so huge that the failure in only one time could be a matter of sadness, at least for me. But the Japanese usually need not to worry about money, at least in good purposes. The Danish company Hexagon made those detection probes depending on the research published on lower species. They could make microRNAs visualized grossly in the whole body. But in the experiments, we succeeded to localize and visualize microRNA-34a in the cellular level and even in different compartments inside the cell. We had special facilities and place for RNA-related research in the department. We succeeded to visualize microRNA-34a in almost all cells in different rat organs. It was known at that point that the expression of microRNAs can vary in different organs. Something interesting got cleared from my experiment that the expression of a single microRNA can vary among different cells in a single organ. The expression of a single microRNA can even differ in different developmental stages of a single cell. At that time, everybody knew that mature microRNAs are produced and work in the cytoplasm of a cell. Even today, in 2021, the same thing can be found sometimes. I myself also knew the same information. But unexpectedly, we found the presence of mature microRNA-34a inside the cellular nucleus or nucleoli in my experiment. We did not have any idea about the functions and importance of mature microRNAs inside the cellular nucleus. We only reported our observations in the research article. People usually get afraid of in case of saying something new out of the traditional thinking and belief. A researcher strongly needs to exclude the influences of experimental artifacts and errors in the first hand. It is essentially needed for a researcher to be confident and accurate about the experimental procedures and practices. Otherwise, a very little wrong information in any published article can cause great harm to a researcher and a research group. In 2019, I was trying to write a summary of my previous research in LinkedIn. I tried to check the details about the nuclear microRNAs and found that Dr. Thomas Sosol and his group in the USA discovered and published the presence of mature microRNA 21 in the nuclear fraction in July 2004, long before my unexpected discovery of mature microRNA 34a inside the nucleus. We were not informed about their big findings in this period. The group in the USA who discovered for the first time the nuclear microRNA 21 were very famous for their works on small RNAs at that time. I myself was also used to keep my eyes on their works in a regular manner and tried to take benefits from their published works. At that time, the concept of cytoplasm-centered functionality of microRNAs was so dominant in my mind, which led me to miss this great information. We actually could not use any supporting reference for nuclear microRNAs in our article. The group in the USA fractionated different cellular compartments and succeeded to detect signals for the presence of mature microRNAs in the nuclear fraction using northern blotting. I could find another article that reported the presence of mature microRNA inside the nucleus before our group in Japan. Joan C. Ritland, Polish et al. Published an article in December 2006 in the Journal of PNAS entitled microRNA 206 co-localizes with ribosome-rich regions in both the nucleus and cytoplasm of rat myogenic cells. They used the in situ localization approach using microRNA detection probe from the same Danish company, Exicon, and succeeded to localize mature microRNA inside the nucleus and nucleoli. But we were not informed at all about their great findings while reporting the presence of mature microRNA-34a inside the nucleus. We also used in situ hybridization approach to localize the presence of mature microRNA-34a inside the intact cells and subcellular compartments. My supervisor was a famous pathologist doctor in Japan. He had huge expertise and knowledge in histology. He had precious research experiences of working in the Food and Drug Administration (FDA) in the USA also. It is to mention here that he was honored with National Award of Japan in 2015 for his contribution in the pathology section of medical sciences. I think that he was satisfied with my experimental performances in his laboratory, and for certain he could realize the long-term impacts of the findings, which may promote the inclusion of this unexpected observation in the article. Because he usually did the final modification and edit of a manuscript for submission in the scientific journal, a supervisor usually wants to be sure about the reproducibility of at least an expected discovery using the same parameters and setups of the experiment. Because we see a PhD supervisor to be famous in scientific world, at least in the direction of his or her research. A famous scientist usually tries to make sure the accuracy and authenticity of the contents of a research article before permitting someone to add his or her name in the author list. Anyway, our experiment may be considered as the first attempt globally. Today we know that a huge number of mature microRNAs work inside the cellular nucleus. It makes me a little bit happy because I invested a huge efforts for the in situ detection of microRNAs in Japan. The connections of nuclear microRNAs in normal physiology and in different diseases has already been explored to a notable level today. For example, cancer, hypoxia, metastasis, cancer-related angiogenesis, regulation of cytokine synthesis, inflammation, asthma, autoimmune disorders, and others. The known functions of mature microRNAs inside the nucleus include post-transcriptional gene silencing, post-transcriptional gene silencing, transcriptional gene activation, modulation of epigenetic mechanisms of gene regulation, and others. The translocation and transportation mechanisms of mature microRNAs from cytoplasm to nucleus has also been explored to some degrees. Anyway, I would like to wish happy, prosperous, and dignified life for you everybody. Thank you.